Good evening and welcome to the Biological Station's fourth public lecture of 2020. My name is Knut Nadelhofer. I'm the director of the Biological Station, and I'm an ecologist and professor in the Department of Ecology and Evolutionary Biology at the University of Michigan. I am speaking to you now from my director's cabin on Douglas Lake at our Douglas Lake campus, which is essentially void of students. All of our classes are being run online this year. But we're pleased to share these lectures with you, not only with our students and our faculty and staff, but also alums, neighbors, friends, and anyone in the world who wants a biological station experience. We are located at the tip of the Mitt region in northern Michigan, about 20 miles south of the historic and geologically interesting Mackinaw Straits. And we will revert to our speaker in a minute. I just wanted to encourage all of you to use the Q&A function at the bottom of your Zoom screen to make comments or ask questions during the talk. Our organizer and host, Ms. Jenny Callis, will then read the comments and questions to our speaker when he is done speaking. Before we proceed, I want to thank all of our students who are taking their field courses online this summer due to the COVID-19 pandemic. That's challenging human health and well-being across our state, across the nation, and around the world. We thank you for joining our program this year, and we admire your perseverance and creativity as you conduct class projects and field research from wherever you may be living this summer. We will work very hard to bring you up to the Biological Station for a long weekend trip if you happen to come back to the University of Michigan, either the Flint or Ann Arbor or Dearborn campus, or anywhere from within the state of Michigan. We can't promise, but we will make that super effort to bring you here for a reunion field trip in real time. I also want to thank our hardworking faculty, one of whom will be introducing the speaker, Professor Charles Davis, for really confronting and meeting the unique challenges of teaching field courses online. I know online teaching is what everyone's doing from K through Gray in the US, but teaching a field course is a particular challenge. You know, we want our students to have experiential learning out in the field with plants and animals and ecosystems, land and water. And our faculty are managing, and their teaching assistants are managing to engage our students. So I want to thank them for that. I want to thank also our dedicated staff members who are working to connect our instructors and our students by providing resources for sampling, mailing materials out, receiving materials, analyzing materials when they come into our chemistry lab, providing advice with data management and writing. So thanks to all of our staff. And finally, before I introduce our introducer, it's important that we acknowledge the state of Michigan. The entire state was appropriated from indigenous people early in the 19th century. The Biological Station operates on 13,000 acres of nearly 14 million acres that were forcibly ceded to the US government by the Treaty of Washington in 1836, 1 year before Michigan became a state in 1837. We're grateful to the Odawa and Chippewa nations and other Anishinaabe people for their many centuries of sustainably stewarding the lands, waters, and resources we now study and share with them. We thank them and we thank all of you for your work. Now I'm pleased to welcome Dr. Charles C. Davis, professor in the Department of Organismal and Evolutionary Biology at Harvard University, who will introduce our speaker. Professor Davis is a Michigan native and a Biological Station alum. He received his Bachelor's of Arts in Biology with honors from the U of M in 1997. He received his Master's in Biology from Harvard in 1999 and his PhD from Harvard in 2002. The year I was actually hired as director of the Biological Station. So I met Professor Davis shortly after he got his PhD. He was an assistant professor at the U of M in EEB and in my department, Ecology and Evolutionary Biology, and a junior fellow at the U of M from 2002 to 2005. So that's when I first met Charles. In 2005, he moved to Harvard, and where he was an assistant professor from 2005 to 2010. He's now a professor in the Department of Evolutionary and Organismal and Evolutionary Biology, excuse me, and curator of vascular plants at the Harvard Herbarium from 2011 to the present. He was director of the Harvard University Herbaria from 2013 to 2019. And now that he is no longer director, I'm sure he is very happy to have more time to spend on his research. As a director who is leaving at the end of August, voluntarily retiring to emeritus status, I am looking forward to working on many other things, and along with Professor Davis, thinking back about all the wonderful things that have happened at the biological station during my career here. So without any further verbiage from me, it's my great pleasure to introduce Professor Charles Davis. Thanks, Chuck.
Great. Wonderful. Good evening, everyone. It's nice to see you all here. There's actually about 150 of you that are out there in the audience. So thanks so much for joining tonight. You're in for quite a special treat. And thanks to you too, Newt, for the very warm introduction. As you know, I've really appreciated and valued both your leadership and your friendship and support over the years. So it's my enormous pleasure to introduce our speaker tonight for a whole variety of reasons, Dr. Anton Reznicek. But before introducing Tony, though, I wanted to acknowledge Dr. Ralph Bennett, who made this ongoing and annual lecture possible tonight. Dr. Bennett was actually a graduate of the University of Michigan, where he obtained his doctorate in botany, which at the time included mycology and microbiology. Those were his specialties. He was also a student at bug camp at the UMBS from 1944 to 1946, and it was during this time that he really formed an early and formative love of biology. His experience was actually in the field of fermentation research, and he was especially involved in the development of penicillin and bacitracin, as well as other commonly used antibiotics. He was also an expert in vitamin B12. Now, he loved the University of Michigan so much that he actually spent his early career as an instructor of botany at the U, and he later then moved to the Squibb Institute for Medical Research, where he spent most of his career from 1955 to 1971. Now at Squibb, Dr. Bennett headed the Department of Microbial Biochemistry and of Chemotherapy, and then directed the Science of Information Department. So several high-level administrative roles that he played in that capacity. And he had a distinguished career. He was a fellow of the American Academy of Microbiology, a fellow of AAAS, a member of the American Chemical Society, a member of the Mycological Society of America, Phi Beta Kappa, Phi Sigma, and Sigma Xi. But importantly, we wanted to present this talk in honor of his memory. Now back to Tony. So I'm going to personalize this a bit for you in the audience because I've known Tony for quite a long time, since at least 1995. Now, the first thing to know about Tony, and perhaps the main thing you need to know, is that he's the world's authority on a group of very difficult plants called carex. These are the sedges. They're little graminoids that inhabit a variety of habitats, including aquatic environments. Now, these are species that you commonly need to study with a hand lens, and they often are required to be sort of at the perfect stage of development to actually know their identity. And all this is to say that they can be quite cranky plants, and they're challenging for students at the outset. Now, you might ask, why would anyone study such a tricky group? And it turns out that I figured this out very early on while I was an undergraduate working at the University of Michigan Herbarium. So, and the short answer to this is that Tony really got bored with all of the other easy things, the other easy plants on the landscape that are conspicuous to everyone else around us. And would you say that's true, Tony? And
Well,
yeah, yeah,
partly,
and
partly.
yeah, partly. And Carrick's, I think it began this grand challenge for him. And his expertise has really grown to not only include the ecology of these plants, but the plant diversity and ecology more broadly of those of the entire Great Lakes region. And he's also come to really specialize in the conservation of these habitats that he's loved so very much. But his focus has often been, rooted in the study of the genus Carex. Now, it's no understatement that Tony Reznicek is easily, hands down, one of the finest field botanists in North America. There is simply no one that I rely on more for information on plant biodiversity in this part of the world than Tony. If I'm taking a trip across the country and I want to botanize, I ask Tony, and I have. And I can recall in vivid detail his instructions on where to find a particular plant growing on a particular ridge in Wyoming, of all places. And any of you in the audience that have had the pleasure of going to the field with Tony know exactly what I mean. Now, more recently, I've had the pleasure of teaching with Tony at the biostation. I spent many hours from Tony, learning from him. And of course, one of the beauties of teaching plant diversity is that, as you start to process and understand, you really place the greenery around you. And I think for any instructor in this realm, it's truly the highlight of student engagement, seeing students being able to understand and extend their inquiry. And Tony does this in such a beautiful way, but I've also benefited from these interactions in uncountable, innumerable ways. I can recall actually first scouting Reese's Swamp with Tony, and he probably showed me 4 species of willow, at least along the stretch of dirt road that I didn't know, let alone previously recognize. And this is Tony Reznicek. If you're in a group of botanists and you don't know, ask Tony is pretty much the motto. Lastly, and before I get on to his professional accolades and we move forward on the talk, I wanted to also say that Tony is an extraordinary gardener. He has perhaps one of the best gardens in all of the Midwest. And I can recall in the mid-90s seeing his garden for the first time. And it's now grown, I think, to the adjacent property, and it's quite spectacular. And when I first entered, there were plenty slipper orchids, and I said, wow, this is amazing. And at that point, I truly appreciated his love of plants. He said, well, I buried a bathtub in the ground so that I could grow these here. And then, I moved to the next plant, and it was hemlock. I said, beautiful hemlock. And he said, well, that's fine, but it's not so much the hemlock, but I'm growing this little Buckley parasite that you could see right here at the base of the plant. Super rare species and quite extraordinary for any of you plant geeks out there. So Tony, simply put, he loves plants, and I am deeply honored to consider him my colleague and friend and mentor. So on to the professional stuff. Tony is Canadian, and he finished his PhD at the University of Toronto in 1978 and immediately assumed his role at the University of Michigan, where he's been active in both the Department of EEB as well as the University of Michigan Herbarium ever since. And aside from being a curator at the UMich Herbarium, a responsibility that I know he loves and respects, Tony has actually served in a number of other administrative roles over the years, including the assistant director of the Herbarium, the director of the Mackinaw Botanical Garden, as well as head of the plant biology group at EEB. He's also served as an instructor for numerous courses, including agrostology. It's a course I actually took with him. Field botany, of course, which I have taught with him in recent years, and a whole host of seminars outside and workshops outside of the UMich ecosystem to both amateurs and professionals alike. He served on many graduate committees, many students that have gone on to become extraordinarily um, 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 respected and in their career pursuits, as well as professional societies. And he's won hundreds of thousands of dollars in grants and authored well over 200 papers. So it's a real pleasure and an honor for Tony to speak with us tonight. And I wanted to extend the welcome to Tony, who will speak on looking for tomorrow through yesterday, what Michigan's flora in 1840 can tell us about our botanical future. Thanks, Tony.
Well, thanks very much, Chuck. I'm a little uncertain as to how all this is going to fit together. I think I'm going to start by saying none of this is from personal experience. So even though I date back a fair ways, I really wanted to show people a little bit about the historical collections in the herbarium and about how we can work with those to actually illuminate quite a bit about our flora. And I wanted to go out on a fairly long and thin limb near the end and put out some ideas about what the changes we've seen since 1840 can tell us about future and what we might do both in conservation and to some degree in research to accommodate some of these things. So let me get started here. This is a sketch by Bella Hubbard, who was Douglas Houghton's geology assistant and the Grand Sable Dunes in 1840. But I'm going to go back a little earlier than that. I'm going to begin at the beginning. Thomas Nuttall. If you want the very first plant collections in Michigan, you have to go to Nuttall. And I want to let people in on a little secret. I bet there are a few people who still remember very fondly Ed Voss. And these three photographs on the covers of the 3 volumes of Ed's Michigan Flora are actually 3 of the primary plants described by Thomas Nuttall from Mackinaw Island, and they represent furthermore the distribution types that Ed was so fond of and taught so well in years of teaching at the biological station. Iris lacustris, one of the Great Lakes endemics, Rubus parviflorus, a western disjunct, and of course the Lake Huron tansy, which is a boreal outlier. So that's the secret. Now, Nuttall did gather collections, but he kept tipping his canoe, and so we have, to my knowledge, only one single collection surviving, Rubus parviflorus, which is he collected it on Mackinaw Island in mid-August 1810. And notice he says Rubus parviflorus lost all but this fragment at the bottom there. So yeah, you know, he was a better botanist than a canoeist. But this is the earliest surviving plant collection. There's his description of it. You know, I still can't figure out, everything sounds perfect about it, and then he goes and says flowers small. He must have got mixed up. But this was one of the earliest botanical works to mention things about Michigan plants, and very interesting Michigan plants. And I'm not going to talk about any others, with one exception, because first, there are only a few, and second, there are very few specimens extant. The one person I am going to talk about quickly is Zena Pitcher. Why? Because, of course, Pitcher's thistle was named for him. There he is. And if you're on Central Campus, you can drive down Zena Pitcher Way or whatever it is. I can't remember. And here in describing Pitcher's thistle, Amos Eaton, mentions first found by Dr. Zena Pitcher on the great sandbanks of Lake Superior, the Grand Sable Dunes from that first illustration. My specimen was collected by Dr. E. James at Lake Huron, from which I made the description, not having seen Dr. Torrey's. Well, you know, Torrey kind of played his cards close to his chest, I guess. So Pitcher, however, also collected very few plants. Here's one of his most interesting things, the trail plant, Adenocolon bicolor. And I'm not really showing it because it's so cool. I'm just illustrating one of the problems we have with early collections. Pitcher's find was neat, but much more important, his personal herbarium contained a large and well-labeled set of collections from the first survey. And I'll let you in on what that all means in a moment. But if we have a look at the labels, you can see there's some real issues right off the bat with people trying to use these older collections to get good localities and good data. Here is on the left is John Torrey's herbarium specimen of this plant, written in his hand. And he says simply, Lake Superior, Dr. Pitcher. Well, you know, that is not going to help you with anything. But if we look at the Michigan sheet with Dr. Pitcher's medical notepad paper there, we see actually that it was collected at Sault Ste. Marie, Michigan, June 24th, 1826 at the base of Coal Pit Hill. That is roughly where Lake Superior State University is right now. So, you know, this is useful. So you need to do a lot of correlating, connecting things up, and so forth. This plant actually was baffling completely to both of them. The genus and species were not described until several years after Pitcher gathered it. So anyway, just an aside. So bottom line, the period from these first collections in 1810 through about 1830 is very typical of early beginnings of floristic knowledge for most areas of the US. Data is so scattered, collecting so incomplete, you can't do much with it. If specimens are lacking, you can't use any of the reports because you have no idea what they were talking about. So virtually impossible to utilize in any concrete way. So why do I think we can do better after, we can do more between 1830 and 1840? What happened in that time? Here's what happened. Douglas Houghton. Douglas Houghton, of course, is memorialized throughout Michigan, Houghton County, the city of Houghton, Houghton Lake. There's all sorts of other Houghtons. And he came to Michigan in 1830. He was basically sent there by Amos Eaton. Amos Eaton was the godfather of Michigan botany, I would say very fairly. And, you know, godfather works in multiple ways. Eaton actually served some prison time for fraud earlier in his career. So he definitely was an interesting character. And he was a hugely influential figure, though, in early botany. And there's a very important connection between Michigan and Troy, New York, and the Rensselaer School. Well, anyway, what are the differences? Here is the Houghton era, 1830 to 1840, and look at the pulse in collections and how there were so many collections that came through then. And after Houghton stopped collecting, there was virtually nothing for decades. So we have a lot of material, and it was a different world, though, in November of 1830 when Houghton came to Detroit. Late in 1830, the delegate to Congress from Michigan, Lucius Lyon, actually recruited Houghton, he actually sought advice from Amos Eaton about hiring a scientist to present public lectures in Detroit. You've got to be kidding me, I said. But in contemporary accounts, there is a note about Detroit citizens who numbered slightly less than 2,000 had acquired a desire to learn as much as possible about the scientific achievements of the period in which they lived. See, it was a different world. Houghton started collecting immediately after he got to Detroit. Here's a fungus. I mean, what else can you collect in New York in November of 1830? But that's not really what got things going. The first big contribution that Houghton made was on the Schoolcraft expeditions to the source of the Mississippi River. These were a more focused and scientifically productive follow-up to the 1820 Cass expedition. Now, we don't like Lewis Cass. He was a slave owner, and he did horrible things to the native people under Andrew Jackson when he served in that administration. But Henry Rowe Schoolcraft was altogether a different matter. He was the scientist on the Cass expedition for all except botany, and then he was chosen to lead the two 1830 expeditions. So how did he and Houghton come together? Well, Schoolcraft wrote that while at Detroit during the winter, I had invited Dr. Douglas Houghton to accompany me to vaccinate the Indians. He was a man of pleasing manners and deportment, small of stature and of a compact make, and apparently well suited to withstand the fatigues incidental to such a journey. So he was a good field botanist. That fits. But Schoolcraft actually noted that he was a good botanist and geologist, and that he should have considered it inexcusable to conduct an expedition into the Indian country without collecting data over and above the public duties to understand its natural history. So here we see a huge regard for science, and Schoolcraft was a very interesting figure and was much more sympathetic, of course, to the native peoples because his wife Jane had native ancestry. So Schoolcraft was a very interesting character, also very important in Michigan for reasons other than botany. So the first Schoolcraft expedition. Well, the center of the state at that time was Mackinaw Island. So they all started at Mackinaw Island and they trundled on quickly. These are Google Earth points of georeferenceable collections. They didn't move that fast. July 1st at the Dead River mouth, that's Marquette. And there he collected Pinguicula vulgaris, another lovely plant of the Lake Superior and northern Lake Michigan and Huron shoreline. Presque Isle, Lake Superior, July 1st, 1831, rock. And there it is, still present. These expeditions likely generated Houghton's love of collecting by canoe on northern waterways. Off he went into the hairy wilds of Wisconsin with a little detour up the Ontanagon River. And after a whole bunch of stuff, including not finding the source of the Mississippi River, he came back to Mackinaw Island via Green Bay. So the 1830 expedition traveled much more quickly so as to be successful because they ran into low water. That's why they weren't successful, and collected fewer plants. Well, let me digress. Look at this interesting thing here. It gives a little insight into Schoolcraft, but also the Ontanagon boulder down below at the lower right was a fantastic nugget, giant boulder of copper rock. And Schoolcraft was incensed that the Cass expedition were wanting to destroy it or at least haul it out of there. Well, it eventually did get hauled out of there. There it is today. Oh, that's a bird. I thought that was a nice touch that Schoolcraft put in his sketch. There's the boulder today in the Smithsonian. It's not on public display. I think where they were going to put it, the floor wasn't strong enough to support it. And there's Schoolcraft's sketch, and it's remarkable how similar it looks. It's the same boulder. The point really is, with this digression, that the Schoolcraft Expeditions also cemented Houghton's reputation as a science. And as a scientist, where that came important was when Michigan became a state. The first Geological Survey ran from 1837 to 1840, and only 28 days after Michigan became a state, they approved this act, an act to provide for a geological survey of the state. And of course, this was really Houghton's background activities that promoted this. Houghton had the ear of the governor, Stevens T. Mason, and they were friends, and was a very influential scientist at the time. So here is one of the very interesting things, an act in 1837 to make an accurate and complete geological survey of the state, which shall be accompanied with blah, 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 and botanical productions together with specimens of the same. Botanical specimens. There is what we're looking at. And even though the biological portion of the survey ended in 1840, this was really a major turning point for Michigan botany. It was Douglas Houghton's concept of how to properly plan for Michigan, newly become a state, to utilize its natural resources for settlement and economic development. It shows remarkable foresight, even by modern standards. There is Cyperus Houghtoni, named for a Houghton collection from these expeditions, although this one is in a field near Pelston. And of course, part of the act was that the governor is hereby authorized and directed to nominate, and by and with the advice and consent of the Senate, to appoint a competent person whose duty shall be to make an accurate and complete geological survey of the state. Well, you can imagine who the competent person was. It was Houghton. And there we have it. The survey was organized. It started out a little late in 1837, and there were two large expeditions that went to the southern part of the state in 1837 and 1838. They collected a lot of material. They also went briefly in 1839 to the eastern and southern Upper Peninsula, and they did some incidental collecting in southern Michigan and by Houghton on the northern lakeshore. So, but these two expeditions in the south were the big things. What was special about the first survey? A number of things. First of all, they were part of a systematic attempt to catalog and voucher the entire flora. They were not random botanizing, as fun as that is. The collections were made over a short period, so they present a really clear snapshot in time. They were all made by the same people who are trained scientists. So this gives a lot of consistency in coverage, no noise due to varying skill sets and interests. The collections had labels with habitats. Again, this is unusual, and consistently applied, even if abbreviated by the same scientists. And the area surveyed was large enough to avoid any variability in floristic composition over time, with disturbance, with all sorts of things that you can have plague some smaller areas, especially really small areas. So I think it was a very special collection. There were huge problems with the labels. I already alluded a bit to the kinds of problems you run into with the specimen by picture, but unfortunately, the specimens were not processed until well after the survey ended. It may well be that the original set for Michigan was lost. The curators and assistants of the time actually followed the practice of rewriting standardized labels with abbreviated data, or even just scribbling the data on the sheets, and often discarding the contemporary labels. This was standard back then, but we really are sad that that happened. And these standard labels often included recording only the county and often not putting in the year, and also crazy things like attributing all the collections to Douglas Houghton, even though he never collected with the survey parties. So there was a breakthrough, though, in really resolving this, a couple actually. One of them was that Zena Pitcher's original set that he got, which came to the National Arboretum, has contemporary labels written by people that were right there at the time. And so this was hugely helpful. Now, despite Zena Pitcher's connection to Michigan and the medical school, and he was a regent of the university and mayor of Detroit, I think for 2 terms, there's an important subtext here. Get your affairs in order before you die, because he passed away and his wife sold his herbarium to a wealthy East Coast collector. So it ended up, fortunately, at the National Arboretum, where it's very well taken care of and beautifully curated, but it's an interesting thing. So here you see a little bit of what I mean here. The 2 labels in the upper right are from Pitcher's personal set. Notice the detail, dates, habitats, dry woods and hills, and even common names and things of that sort. And then a lot of the standard rewritten labels say Ann Arbor, June 20th, 1838. St. Joseph, August 6th. That's not helpful. Mason, Michigan, even less helpful. Monroe, June. Crazy stuff. So this is one of the huge problems in utilizing these old collections. And fortunately, with putting everything together, with being able to support it, sort it with modern databases, and being able to do itineraries, it's actually possible to get a lot of good data out of this, notwithstanding. And here's some more labels with some interesting aspects. Squaw Island, we still don't know where Squaw Island is. Luckily, we know where they were on June 16th, so it couldn't have been too far from Roseal. And there's a label with an interesting habitat. Dry oak openings, July 1833, Detroit. Wow, that's interesting. I'll elaborate on that. One of the big breakthroughs before the National Arboretum set was studied was Rogers McVaugh's 1970 historical account. It was pioneering. He was the first person to actually pull everything together, put things in the appropriate year, and work out an approximate itinerary. I'm amazed at how he was able to do that without modern databases and computers and sorting it. It's just a fantastic thing. 1837, both botany and zoology were handled by Abram Sager. There he is in full sideburn. And he did collect a credible number of plants. Rogers McVaugh saw about 160. With examination of Zena Pitcher's herbarium, we now know of about 270. Enough to actually make a nice story. But he was a terrible handwriter. June 16th, Roseal River. That fits for a Potamagiton. That may be an original field label. There are very few of those. So if you whip out Google Earth and put everything on there, you get this nice mapping, and it's really hard to figure out what this means. But for a little context, here is a nice map of Michigan, the map that all well-equipped travelers use, the tourist pocket map of Michigan, exhibiting its internal improvements, roads, distances, etc., by J.H. Young, 1835. And we're pretty sure that the first survey had this map in their back pockets because the place names and even minor localities all correspond. And so giving some context, because this had the stagecoach routes and everything, we can now see a little bit more detail. They started in Detroit, off to Brownstown, Gibraltar, Grosse Ile, took the stage route presumably to Monroe, over to Dundee, Adrian. We don't know exactly how they got to Jonesville. There would have been a couple of choices. Off to Coldwater, visiting the Sturgis Prairie, White Pigeon, and then the St. Joseph River at the southeast corner of Cass County. Then they didn't collect anything at all until Kalamazoo. Why? Probably because they took a boat up the river and may have been perhaps separated from their equipment. So they went to Kalamazoo, and then they hightailed it back to Detroit along the road, which was already well established between Detroit and St. Joseph, Michigan. And boom, there they were, back to Dearborn, back to Detroit. Now we have some context. We have how they traveled and where, and we can do some things with that. I want to point out this map actually has some interesting ecological information. There's Gull Prairie in Kalamazoo County. Kalamazoo Prairie, now Grand Prairie. Well, it doesn't exist anymore, but it was. The Sturgis Prairie, White Pigeon Prairie. And if we look closely, for example, at St. Joseph County, there's the pre-settlement map. There's the Sturgis Prairie. There's the White Pigeon Prairie. And this prairie up here was Flowerfield Township. You'll see the significance of that in a minute. Now, 1837, 270 or so sheets, that's pretty good. But over the winter, the first survey was reorganized on a much larger, better financed scale, more true to Houghton's vision. Abram Sager took on only zoology, and John Wright was hired to be the botanist, and George H. Spull was hired as the botany subassistant. I'm sure glad that subassistant title doesn't exist now. And from this year, with the examination of the set in Pitcher's Herbarium at the National Arboretum, almost all with contemporary labels by George Bull, we have actually more than 800 individual collections from 1838. That's starting to get significant. George Bull had lovely handwriting, and there's Carex limosa, Sphagnus swamp, Northfield, Washtenaw County. That's probably Mud Lake Bog. On the other hand, John Wright had lousy handwriting. What on earth does this say? It says margin of St. Joe at Niles. That's the St. Joseph River. People who recopied the labels, you remember I mentioned that issue, sometimes assumed that St. Joe was St. John's, resulting in strange anachronisms. The town wasn't founded until the 1850s. And also, these kind of things messed up the itinerary. But we got it all worked out. John Wright was an interesting character. I was never able to find a portrait of him, not yet anyway. But he was an established Troy doctor and credible botanist. There's his Flora of the vicinity of Troy, catalog of plants growing without cultivation. And this was 1836. And it was interesting that he was lured to Michigan. And I would suppose it's for two reasons. One, the draw of the frontier. And also he was encouraged surely by Amos Eaton, and I'll show you why. Like Houghton and Sager, Wright attended the Rensselaer School, and so surely Houghton and Sager and Wright all knew each other. And after the first survey, Wright went back to the Rensselaer School, serving on the faculty until a year before his death. Now, this was fairly well known. George Hirambo was a total mystery to both Rogers McMahon and Ed Voss, with the sole exception that Bessia Bulli was named for him. And I wasn't able to find a picture of him either, but there's his father, Archibald. He's a fierce-looking character. No wonder young George went off to Michigan as soon as he could. But later he went to back to Troy, became very active in the community, and actually got his medical degree, moved to California to practice medicine. He married a wealthy young widow in 1851, but sadly passed away in 1854. But his son, George P., became a prominent local citizen. So we know quite a lot about him, thanks to some friends who are whizzes at genealogy. There is where the 1838 party went. You can see it's rather complementary to the 1837 route. 1839 was a bust. There was a huge economic collapse in the US. The restricting of the survey due to financial difficulties was inevitable. But Abram Sager, of course, was fairly bitter about it, commenting, these resignations were provoked by ignorant criticisms and caricatures indulged in by members of the legislature in public debates on questions connected with the survey. So politics hasn't changed much. Bull collected on his own a little bit in 1839, but mostly he assisted Houghton with the trip along the northern shores of the southern shores of the Upper Peninsula. His best find, the type of Solidago houghtonii right there. So very interesting. The end came in 1840. The botanical and zoological departments were abolished. Houghton and Bella Hubbard collected a few plants along their trip to Lake Superior, including a August 3rd collection on Isle Royal, the first plant ever collected on Isle Royal. And this was the trip that Grand Sable Dunes sketch was made on. So Houghton was clearly pretty disappointed by the collapse of the biological parts of the survey. He never collected a plant again during the rest of his life, which was very short. He died tragically on Lake Superior in 1845. Here is his plaque on his monument at Eagle River. But this isn't all that happened with the survey. John Wright somehow managed to hurriedly put together the first plant list for the state in 1838, published January 1st, 1839. And there he complains about the legislature obligating him to collect 17 specimens of each kind. Oh, that is onerous. I don't know where they came up with that number, but they actually gathered apparently about 9,000 specimens, although very few extant collections have more than 2 or 3 sheets, leaving a bit of a mystery as to what happened to all these. So, and there are a lot of specimens that are on a lot of species on this list not represented by specimens, and vice versa. Rogers McMahon was completely baffled as to how John Wright could have done this list, but if we, I happened to see on a label of a Carex that Wright thought was undescribed. It wasn't, but he thought it was. an interesting comment. See notebook, page 259. I imagine that Wright used this notebook in compiling the list. No idea what happened to it, although I have to admit I haven't looked at the archives at Troy. I looked at the state archives and it's not there. What about the collections? Well, in one of Asa Gray's letter, he noted that on a visit to Ann Arbor in August of 1838, you remember at that time he was the botanist at the University of Michigan, professor of botany at the University of Michigan. But Houghton was a pack rat. His home was entirely occupied as a storehouse for the stuff collected in the state survey. Wow, what did his wife think? In February 1843, this was also before the specimens were turned over. There was an inventory noting 15,000 plants. The formal transfer of the specimens didn't happen until 1846, but we don't know actually how all this worked. Duplicates, of course, were important, and that's why there are collections to look at from the first survey at Harvard. There's a collection from Asa Gray's Herbarium. It says Michigan State Collection, very helpful. Others, John Torrey's collection, Shore of Lake Michigan near St. Joseph, that we can place. Another one, ex-Herb George Thurber at the Gray Herbarium, Mason, Michigan, collected by Houghton. Pretty poor, but we have some idea of where most of the sets went. Basically, 3 herbaria have them all. 4 herbaria, Mich, New York, the National Arboretum, and Harvard. So there's where to look. Now, what's the other reason Wright went to Michigan to collect? It may well be Amos Eaton's urging to expand his North American botany. And here is Wright's magnum opus, the Eaton and Wright Manual of 1840. And there they note lots of records by Houghton, Cooley, Edwin James, and records with MCH. Behind them are from the first survey. And there's an example with Pitcher's Thistle. And unfortunately, Eaton didn't keep specimens, but we won't hold that against him. Even though he'd quit, he still had on the Flora state botanist in the Geological Commission of Michigan. This came out in 1840, so that's the other reason for the endpoint. There's where they covered, not bad. I mean, missing huge chunks of the state, but it's very fortunate that they focused on the southern tier of counties, which were both very biodiverse and also developed quickly. So what can we learn from all this collections? About 650 collections have habitat information. It's kind of puzzling. There's a collection of a sedge from Ann Arbor, damp openings. Damp openings. What on earth is an opening? Fortunately, we have contemporary write-ups. Bella Hubbard. The ordinary character of the openings is that of a majestic orchard of stately oaks, which is frequently varied by small prairies, grassy lawns, and clear lakes. These magnificent groves were, until within a few years, kept free from underbrush by the passage through them of annual fires, allowing successive growths of herbage to spring up luxuriantly, covering the surface with a profusion of wildflowers and verdure. They loved these places. And it gets even more effusive. So we see the firsthand account here of Native American land management with fire until close to the time of these collections. These openings were very nicely described. And he wasn't the only one to be impressed by oak openings. There's the novel, James Fennimore Cooper, 1848, set in Kalamazoo. There probably were still openings then. They may have looked like this. This is a scene from Walpole Island, Ontario. Why do I say that? Walpole Island is unceded Native American land that was never subjected to European land clearing or agriculture, but where fire management was maintained until the present. So this may well be a nice view of it. And here's another area that has been recovered at the Black Oak Savannah at the Ojibwe Prairie in Ontario. And this also may give a hint of the area and the beautiful wildflowers. So we have tiny little scraps left. If you list all the species, that's probably a pretty good list of some of the major and conspicuous plants of the openings. There are funny things about these things too. A lot of plants with habitat woods or dry woods included things that we now consider more typical of open habitats. Dicanthelium liebergii, Elymus canadensis, Lilium philadelphicum. Woods? Surely not. But if you look at typical rich forest understory species, Allium tricoccum, shady woods, Hydrophyllum canadense, deep shady woods, Panax quinquefolius, ginseng, deep shady woods. Shady woods would be redundant now, but it probably was not in the 1830s. The woods then were doubtless really more open, and that's of course again because of fire. And the true prairies, in fact, are completely gone. There's really nothing left of them whatsoever. Houghton noted in one of his 1840 reports, the prairie is one wide, unbounded highway where no obstacle is present for pursuing whatever course curiosity may direct. And they restricted the term prairie, exactly as in the modern sense, to the large true prairies in southwestern Michigan. These, unfortunately, of course, among the first tract to be utilized by settlers. The list is small because most of the typical prairie plants they would have got earlier in the season on the way to western Michigan in the openings. And of course, it's interesting that there are also a few aliens there. The prairies were already being disturbed. Now, a few highlights. There were a surprising number of species collected by the first survey that were extirpated, including at present, including quite a number that were never found again. They were collected by the first survey expeditions and never seen again. And this is actually pretty interesting. Many of them are prairie plants. Most of them are prairie plants, and many of them give some clear habitat ideas. Agellinus auriculata, dry openings, bur oak openings, sandy oak openings. There they are. You know, kind of cool plants. Everyone knows Echinacea. So what do we conclude from that? Well, all these extirpated species were from southernmost Michigan. No species collected up to 1840 from the upper peninsula or northern lower peninsula are extirpated. They actually represent about 30% of the native plants recorded as extirpated from southern Michigan. None of them were closed canopy forest plants. Only 3 were obligate wetland plants. That makes sense. It was too early for extensive wetland drainage. But a couple of other interesting points, one especially, not all of these were exceedingly rare plants. Buchnera was found 3 times. And Agellinus, Echinacea purpurea, and Scutellaria incana were each found twice and then never again. So, you know, there were some enormously rapid declines, presumably due to the double whammy of habitat destruction and fire suppression. A couple other sidelights. There were a number of exceedingly rare plants, rare in the modern day, that were collected repeatedly by the first survey. I don't think this was done because they were excited to find them. It was probably to fulfill their mandate of collecting 17 duplicates of each. The prairie white-fringed orchid, Platanthera leucophea, they encountered that 4 times. I mean, now it's inconceivable that you could encounter that 4 times in 2 short summers without looking up known sites. Sebacia angularis, a rare gentian, was found, was collected 6 times. Wow. And we do know from a number of avenues of evidence that things like orchids and gentians are among the first groups of plants that turn up missing from fragmented, disturbed sites or urban woodlots, little patches. There may be a phylogenetic signal to all this, and probably based on this weak evidence, this occurred even before climate change impacts, which show even stronger phylogenetic signal. But I also suspect, though with no evidence, that the smaller or more disturbed the site, the stronger this signal will be. Of great interest to people was that a lot of boreal plants were found by the first survey. And this seems interesting because many of these disappeared very quickly, well before climate change started warming things up. McMahon listed a number of them in 1970, but he missed the most spectacular one, Herbarium vulgaris. There it is, the sole Southern Michigan record from 1838. Even if you find this now around the station area, it's cause for mild excitement and minor knee weakening. But down there, it's spectacular. So that's interesting. And there's a Carex. I had to put a Carex in here. Carex tenuiflora. If you look at a map, 2019, it's still extant above this red line. Below that, all the records are old. I'm cheating a bit here because this was actually not found in Michigan until 1845, but you know, there can't be any doubt it was there before then. But I was intrigued by this 1961 record because that came from the George Reserve, the University of Michigan property, where both the uplands and the wetlands were really quite undisturbed. And sure enough, it was still there. It took a little bit of looking, but it was still there. So now all but one record south of this red line are 100 years old or older. So there are some interesting things. The one record farther south was from a spring and seep complex on the Grand River in Jackson. And this is now the site of Jackson's drinking water treatment plant. They probably utilized the wells early on and utilized the springs. So we have a few things. The use of the word prairie by first survey scientists was really precisely in the modern sense. They were the first and sadly the last to experience true prairie as a landscape ecosystem in Michigan. Combining plants with openings or prairies gives a reasonable qualitative picture of the grassland flora of pre-settlement Michigan, I think. Much of the upland area of southernmost Michigan was a fire-generated savanna, but it's not going to be easy, maybe not possible, to untangle closed canopy forest versus savanna. Most extirpated species found by the first survey were prairie and savanna plants. All forest species collected by the first survey are still with us. They collected a lot of northern species, especially of wetlands, a few of which were never found again in southern Michigan, though of course many are still frequent in the north. And by 1840, aliens were already about 7% of the flora. That may seem awful, but the current figure is 38% and climbing. So, rarity and extirpation in the mid-1800s, total habitat destruction for prairies, well, obviously, fire suppression. But this doesn't answer the question of why boreal species diminished well before any strong climate change impact or extensive drainage. And I think that so many of these species, the clue to their persistence was probably they occurred in localized, tiny cold spots, groundwater springs, groundwater seeps, perhaps also in wetlands that had a lot of cold air drainage. And these microhabitats were very susceptible of disruption. And in particular, whenever there was a spring near any habitation, it was typically converted to a well. They would dig a box and convert it to a nice year-round well. So, and I think that this microhabitat idea is a clue that may give us an insight into why many plants, even beyond wetland species, disappeared so quickly and even disappear so quickly now, because most of these microhabitats are little spots that wouldn't show up on large-scale surveys. And it not only makes finding them difficult, but it also makes them very susceptible of extirpation locally. So I wanted to emphasize one last bit. These first survey collections are uniquely valuable because there are so many of them. They have depth and breadth and consistency. So it really was worth the considerable effort by a number of people to upgrade them so they're fully utilizable for science, and they are available to everyone. Because not only are they usable for early floristic records, itineraries, but with care you can use the data to add a floristic component to our pre-settlement vegetation data, and we have enough that some of them can be used as physical specimens for baselines for analyses requiring actual material, secondary metabolites, isotope analyses, all manner of things. So I think that this is a collection that could be of considerable value in the future still. Now, what about the future? Well, one of the saddest things, of course, is we're piling on lots of new reasons for plants disappearing without ameliorating any existing reasons. Large-scale wetland drainage was a huge thing in the 1930s with the Civilian Conservation Corps. Besides planting trees, that's what they did. Invasive species were really starting to become abundant in the middle of the last century, rising nutrient inputs. I'm not even sure when that hit. Pollinator declines, linked perhaps with dramatic increase in pesticide and herbicide use. Deer and climate change probably from the 1980s on. These are all piled onto habitat destruction and microhabitat degradation. Even though, of course, all you have to do is drive around southern Michigan and look at all the building and you'll see habitat destruction is still a huge thing. I think one lesson is more attention needs to be paid to microhabitats, microhabitat diversity and maintenance, protecting and studying springs, seeps, and vegetation that is supported by them, fens, and generally our water resources overall. And I'll be intrigued as we venture into the future to see how climate change is really impacting things. I wonder if it might be stratified, affecting northern species and then perhaps operating more strongly in the northern part of the state where habitat destruction is operating less. And of course, also, how will species that can't stay in sync with pollinators, what will they do? There's so much there. And that brings me to, I think, the last slide, the broader picture. I think, you know, too much change has happened since 1840 to utilize these older data for study of more recent change. We typically don't know when the disappearance or other shift actually occurred. A problematic corollary of this is that we generally lack ideal baselines, ideal measuring sticks to look at change. We make do with existing data as best we can. I think, you know, we can do something about this perhaps. Floras probably should be constructed more in terms of snapshots in time rather than cumulative as they are now, and to accommodate scale. In other words, scale them to different regions so we can see how change occurs differently in different size areas. We need modern and repeated every however many years, 10 years, 20 years, snapshots of large protected areas or stable subsets. I mean, ideally this would include national parks, but of course they don't like too much collecting for documentation. But it certainly could include some university properties. That's a hint. And I think where feasible and appropriate, we really should collect herbarium material, especially of commoner species, because we'll want to pick apart that for destructive sampling in the future too. Who knows what analytical techniques will be developed in 200 years. And I think just more philosophically, the short time frame we look to with modern science also kind of impairs thinking about baselines whose development is kind of, you know, in some ways unscientific, in some ways not. But of course, it's hard to support with granting agencies. And also, you know, we have it so easy compared to the pioneers of Michigan botany that we should make every effort not to waste the opportunity we have. And here I'll end with a couple of collections from Harvard whose labels need upgrading. So at that point, let me see about any questions, and if anything wasn't clear, or and I'll turn it over to, I guess Jenny is going to take care of the session.
Yep, that's right, Tony. I'm tuning in here. First of all, I just want to say thank you. I'm absolutely blown away by the level of detail. And I wrote a note here, investigative botanizing. That's mine. So really impressed, learned a lot. Thank you, Tony. And I'm gonna go ahead and, without further ado, read some questions that are coming in. So I encourage everybody to continue to ask some questions in the Q&A function. So first off, Tabitha is asking a question. Tabitha is our very knowledgeable ethnobotany teaching assistant at the station. So Tabitha asks, for the extirpated plants of southern Michigan, would it be possible any seeds or other remains would be charcoalified from the annual fires that maintain the prairies? Could we find any kinds of remains?
Wow. It's not inconceivable if we sampled bogs in southwestern Michigan, and there are some. Perhaps pollen is a little more likely, but the answer is yes. I don't really follow that literature. I'm not exactly sure how many southwestern Michigan bogs have been sampled, but I think it's conceivable.
Great. So a question from Kathy. I would like to learn more about those phylogenetic signals that you refer to with the orchids and gentians.
Oh, well, I'll pass that to Chuck. He's the expert on that. And he's done some wonderful work with looking at Thoreau's notes on phenology and collection of and plant occurrences in Walden and surrounding areas, and has had some really interesting results come out of that. Is that fair to ask somebody else to answer your question?
Yeah, totally. I'm happy to chime in. Tony, awesome talk. I had a question too that I added to the chat window here, but just great. So I would just add for Kathy's question that, yeah, so one of the most sort of important sort of climate change response traits in plants, at least the most well studied in many ways, is their phenology when they're the timing of their life cycles. And so this may manifest by plants producing earlier their flowers earlier when springs are warmer, and what we found using these historic data that actually traced back to Thoreau, who kept quite detailed notes on these sorts of phenomena, is that there seems to be not only a striking sort of phylogenetic association between those species that respond dynamically to temperature in terms of their flowering and those that don't, but also a strong association with their change in abundance over time. So we know that climate change is, at least in this one community, is not plants are changing in sort of a non-random or non-uniform way, such that the non-responders, like orchids and plants like that tend to be linked through close relations and are not as dynamic in responding to climate, are the plants that are lost. So there seems to be striking and strong phylogenetic associations. So Tony, I hope that answers Kathy's questions. But again, great talk. Thank you so much.
Yeah, I mean, we definitely see orchids and gentians disappearing. I mean, that much is that comes through in other data as well.
Yeah, and there's certainly other factors involved, as you so nicely pointed out. I would say that to the best that we could, we tried to include those in our models, including even deer browse herbivory. But suffice to say that certainly there are other factors impacting these changes in abundance, not just climate change response or non-response.
Great, thanks for jumping in there, Chuck. So another question here from Dan. What was the prevalence of coastal plain disjuncts among the collections, and are there detectable changes to this floristic element of sandy, wet shores?
They went through areas where coastal plain disjuncts occur, and they found them, and they didn't get very many. They got some of the classic species like some of the sedges like Furina pumila. On the other hand, they didn't find everything. They missed some species that I would be sure they'd almost have to walk over to go and gather the Furina. So, so it left me puzzled as to what communities they were getting these from. But yeah, these were lake shores. They visited Austin Lake in Kalamazoo, which even today is a classic coastal plain disjunct locality, and that's where they got several of the things. But they found others elsewhere as well. They certainly picked them up. They were there in numbers.
Great. So Rex, our algae professor, wants to know, what were the vectors for the early arrivals of exotics?
That's a very interesting question, and I looked at that, and there were two primary vectors. or two primary sets of early exotics. One was medicinal plants and herbs, kitchen herbs. There were a bunch of weird Solanaceae that were around then that no longer are found now. They were being grown surely for medicinal purposes. So that was one group. There was a second group that were routine grain field and garden weeds of Europe that almost certainly came in with contaminated seed. Seed cleaning was not very well developed then. So those were the two primary groups. They were all associated with disturbance, roadsides and farm areas. There were no invasive plants that were going into native ecosystems. Also, there were no escapes yet of plants that would seemingly have been cultivated solely for ornament. These were medicinal plants and grain field weeds.
Okay, so question from Melissa here. This is kind of more a forward-facing question. Hi there, big fan of Michigan Flora. Curious if the Seeds of Success program can be of any help in any borderline rare populations that might continue to disappear under climate change, or reintroduce in potentially good restoration microhabitats.
Yeah, I think so. I mean, I think trying to protect our native genotypes of Michigan rare plants is a win-win situation. Absolutely.
Great. So another question, what tools do you suggest to use to help target those more sensitive sites to find seeps, springs, etc. on a landscape scale? Are there geospatial tools that can help with this?
Wow, well, that's getting a little beyond my zone of expertise. I would say many of the sites are linked to river valleys, and especially river valleys that have associated fens. I think that they are tightly linked to moraines and caves. I think because the gravel, of course, was the conduit for the water, and you know this I think is important too for conservation. You can't protect a fen and then have some gravel mining company strip all the surrounding hills down to the water table. So you know, you have to have these things together, and I think these small scale areas were very quick to be disrupted. But in terms of searching them, that's a good question. Yeah, I didn't comment on it, but there were other even rarer microsites that Houghton himself looked for, and those were salt springs. And of course, that's because of the importance of salt in the early economy of Michigan. And so these salt springs are even rarer than some of the routine mineral and cold water springs and seeps, and almost none of those remain extant. Only one good example remains extant. So, but there's such small patches that it may be that a hydrologist would be able to help focus some geological or other clues that would help turn those up and search for them.
Okay, calling all hydrologists. So another question from Manu, what role do bryophytes play in the herbarium and in historical investigations? And are there plans to make these collections available to the public on Michigan Flora online?
Well, not on Michigan Flora. Because of our expertise deal only with plants with backbones, vascular plants. But the bryophytes are available through the Herbarium website in a reasonable searchable form. I mean, just as an aside, there are, the first survey did apparently collect some bryophytes, but as far as I know, none survived, at least not in Ann Arbor. But yeah, they should be mostly available through the Herbarium website, through our databases.
Great. Another question, what kind of collaboration is being done with Michigan land managers to use this research to influence current management techniques?
Well, of course, the classic example is the reinstatement of fire. And this is being done at a range of scales all over the southern part of the state. And beyond that, I think there's a little less, but fire management is the obvious thing. And of course, this came about well before our knowledge here. I mean, the fact that Michigan had so many fire communities was well known, but you know, my small contribution is just linking the floras and linking contemporary accounts by scientists to this, rather than a lot of the anecdotal or presumptive conclusions.
Okay, so interesting question for the modern era here. Is it still important to actually collect physical samples of plants? I feel like collecting identification type photos can provide more realistic understanding of how things look and might be better. What consideration should someone be thinking about when deciding some plant is important enough to collect a physical sample?
Yeah, that's a really good point. I mean, I think at a very coarse scale, you can't get DNA out of a photo. And you can't get other things like pollen or anything that is linked to actual tissues. But of course, for very rare plants, a photo, perhaps with a diagnostic part, would serve as a voucher, but it wouldn't be very useful in the long term. I think plant collecting is often a little misunderstood in that, you know, for the most part, with a few exceptions, we don't kill plants when we collect them. I mean, there are exceptions, but, you know, when I collect sedges or trees or bushes, I mean, you can't even tell I've been there, even if I've collected a number of duplicates. So I think that you should collect to get the maximum information. You should do it in a way that doesn't hurt the plant. If you have to hurt the plant, then you start thinking about other considerations. So, but often, of course, populations are very large and there's really no discernible harm taking a few. So in general, it's basically more or less the equivalent of, you know, mushroom hunting or berry picking. It's, you know, it's generally not harmful at low scales.
Okay, so question here from a former student. Hi, Gigi, thanks for asking a question. So she asks, when the biodiversity of a landscape changes due to long-term stoppage of the fire treatments by the land's Native American stewards, what is your opinion of the path forward? Should we return to fire treatment if and when possible, or should we let the different ecosystems emerge? Where do we draw the line between natural systems and human influence systems?
Wow. Yeah. I mean, basically, what you're asking is, do we try to have some sort of a set point that we use as the standard for Michigan communities, or do we just
Sorry about that.
Yeah, I think that the answer should be a couple of fold. One of them is, of course, it's just simply not practical to utilize fire everywhere. In many communities, they may have value that has evolved in 200 years that would be disrupted by burning. But I think in the same element, we do view, for example, our national parks, our state parks, for example, in some ways as museums. And I think that thinking in those terms might help make some decisions. I wouldn't go all one way or the other. I think we have to make some decisions about some natural areas. Definitely.
So we're running out of time here, but I want to squeeze in a couple more questions if possible. One is from Chuck Charles Davis, who introduced Tony. I think it's a great question. He asks, it seems that these early explorations heavily dictated the course of subsequent botanical exploration. I think these sorts of efforts in retracing our past routes create biases that can be seen in collections even to present times. In what ways did retracing similar paths slow botanical explorations of Michigan? Were there areas left untouched that in hindsight would have been better explored?
Oh, absolutely. I mean, I think there were, I think this was less of a problem with the more in-depth surveys in southern Michigan, but there were problems there too. For example, they went out rather late in the season, and the reason was they really couldn't get out any earlier. The roads would be impassable. But for example, all of northern Michigan is represented by collections only along the lake shore. It's an enormous bias, a gigantic bias. And it would be wonderful to know, for example, what was in some of the areas along, for example, the prairie and savannas along the Wisconsin border in the Upper Peninsula. They're a total blank. And so there were lots of that. And I do think it's true. I think botanists do tend to visit areas where they know interesting plants are. And so they go back to the same old places again and again and again. And that's, yeah, I mean, you see that with everyone, and it's really hard to break out of that, because if you do, you might come home empty-handed, or you might discover something spectacular, but you're not sure of finding interesting stuff like you are if you go to one of these great places like Presqu'île on Lake Superior or the Grand Sable Dunes, where everyone is collected. Yeah, absolutely. It's huge biases, and that's what makes things difficult for interpreting the data. We have so many gaps.
Well, I think this is a perfect last question here. Ruth asks, what is your favorite plant and why, if you had to choose just one?
Oh my gosh. I'll tell you, it is a plant that was pictured there, and sad to admit, I think it probably is that prairie white fringed orchid, Platanthera leucophea, for several reasons. You know, one of them is it's our showiest orchid, and my very first interests when I was ignorant and young was orchids. So many people get started on them, and I still have a soft spot on them. I still think there are lots of research questions to do with orchids and their disappearance. I think there's much more to it than even meets the eye. For example, just try going into a cedar swamp now that in the 1950s might have had 20 or 30 orchids like Reese's Swamp, and see if you can get the total past 4 or 5. There's some dramatic things going on that really aren't even on anybody's radar screen. And besides, there was a Platanthera hybrid with Leucophia as one parent named for me in Canada, which I happened to be the first to find. A minor thing.
Wow, that's incredible. And I think that's a great spot to end. So first, I'll thank everybody for asking questions. I hope we got to most of them. And Tony, I want to thank you. That was absolutely fantastic. We really appreciate it. We're getting tons of comments here. Everybody really enjoyed it and appreciates your time. So thank you so much, Tony, for this great talk.
It's my pleasure. I had a lot of fun doing it.
All right, I'm going to end this meeting now, but just so everyone knows, this is recorded and it'll be posted on the UMBS website. So you can relive the investigative botanizing over and over again. So thanks everybody for tuning in, and I hope you all have a good night.
Bye, everyone.
Bye.
Bye-bye, Tony. Thanks again.
Bye.